exhausted. But I'm here for you. Say hello to Presley Ray. Don't mind me while I eat some French fries. We skipped dinner, so.
And don't forget to give me French fries.
Mm-hmm.
Thank you.
All right. I can see that some of y'all are in. Go ahead and comment off to the side so I know the commenting's working. Who? Oh, heavens to Betsy. When you don't put your actual name, it makes me not want to answer your questions. Shish dibnigna, whatever you are. Put your actual name. That way I know who I'm talking to.
And who she's
That's just kind of like, you know, the mature grown-up thing to do. Hey, T. Okay, commenting is working for others, Lauren. I'm pretty sure the issue is if you try and do it from your cell phone, commenting won't work. You have to be on your computer to comment. But if you can't be on your computer, you can just send me a text through Remind and I'll try to answer it. But if there's 50 of y'all trying to send me reminds, then I won't get to them.
only.
Okay, Patrick, I see you're commenting. Are you saying that it won't work on your Chromebook as in you can't see the video or you just can't see the comments?
Mommy, can I have some more French fries?
Helen. Won't even open. Patrick, how is it not opening on the Chromebook, but you are commenting on this? All right. Okay, I'll talk about microtubules in just a second. So, Lauren, I don't know if that's the issue for you. You're on your phone and it's working, but on your Chromebook, it's not. Nothing really I can do about that unless I was physically looking at your Chromebook. It's just a YouTube live. So unless they have it blocked, like, is it telling you that it's blocked? Like those warning web things that they put on there from tech? If it is, then that's something I can put in and make sure that they don't do that again. Ah, it blocks chatting because of these safe search filters, I'm pretty sure. Okay. I'll reach out to tech and tell them, you know, stop it. And I'll do my best to tell them that. All right. Microtubules. So your cytoskeleton, one of the components of your cell organelles that I want you to know, your cytoskeleton comes in 3 different categories. Well, I'm trying to find my pen. Where's my pen, Presley?
I don't know.
I got it. So you either have microtubules, microfilaments, or intermediate filaments. It's essentially like saying with your bones, you have hollow bones, flat bones, saddle bones, joint bones, so on and so forth. Like you have all these categories of bones in your body. A cell has these categories of skeletal proteins. So we call microtubules them because they are actually kind of, a microtubule is kind of hollow. So just imagine, you know, like a water hose, and then it's got the proteins, you know, that create this kind of circular structure. But in the center, it's all hollow. So microtubule is simply just a type of protein, and the actual protein fiber that makes it up, we call tubulin. All right, so a microtubule is a type of protein category, right, made of a protein fiber called tubulin. This is one type of skeletal protein that makes up a cell. Then you've got microfilaments, which use things like actin. So a microfilament is the type of protein, and actin is its kind of protein fiber. And these twist around each other. Okay, kind of just think like a braid. So as one goes down, another actin fiber goes down around it. It just kind of braids, twists around like a piece of rope. And then you have what's called intermediate. An intermediate filament, they kind of help out everywhere. Your two main ones are the microtubule and the microfilament. Microtubules are going to be in charge of cilia and flagella, so movement. Okay, they're also going to be in charge of extensions, just sex pili and stuff like that. Your microfilaments and actin, those are important when we're talking about mitosis. So you'll see that again when we get to mitosis. Right now, you just need to know, it helps with cell division. All right, you don't need to be too concerned about the how. Intermediate, they kind of help everywhere, but the kind of protein fiber that's helping there is keratin. All right. And again, that's just pretty much any kind of cellular structure, any kind of assistance, moving vesicles, stuff like that. Intermediates can be there. But your two big hitters are microtubules and microfilaments. All right. Good on the microtubule. Are we going to have to draw and label anything on the test like last test? What did we have to do on the last test? I kind of don't remember. Oh, yeah. Triglycerides. Oh, yeah. You had to do that. It is always an option, especially in biology, that you'd have to draw something. So in general, the things that you would have to draw, like what I would do is I would give you some kind of structure to the drawing. Like maybe I give you a phospholipid bilayer, but I want you to draw in the proteins necessary. Okay. Or maybe I give you this and I want you to draw in where you would find a glycoprotein and a glycolipid or something. You might also get things like, yes, I give you a general cell and I've given you the nucleus or something, and I want you to add in the rough ER, the smooth ER, and the Golgi or something. Yeah. Any component where there's drawing in here, you want to be able to draw a very basic structure. I understand y'all are not AP art students by any means. Okay, so it's not too crazy. Hold on, I'm getting comments on here and in here, so just give me just a second. Do you have the worksheet with you so I can ask you questions off of it? I don't have it in front of me, but I can try to. Hold on. Olivia, can't figure out how to comment. Olivia. Okay, signal transduction pathway. I see that. Olivia, if you're on your Chromebook, it has commenting turned off. I'm guessing, though, if you can work on your phone, it works there. Okay. All right. All right. So don't keep asking too many questions just yet. Let me get to some of these. All right. Tatum, I'll pull up. Give me just a second. I'll see. Phil, let me pull up without interfering with the thing. I'm pulling up the worksheet for those that were needing to look at that. Some specific questions. So hold on. All right. Lauren, you specifically want to know about number 17. It's the proton pump and sucrose. All right, so anytime you have any kind of transport with the cell, it can either be passive or it can be active. Passive, no energy is needed. Active, ATP is needed. But when we're looking at an ATP needed transport, so a type of active transport, what that literally means is that ATP is going to be broken down. It's going to be hydrolyzed, and you're going to lose a phosphate. So it turns back into ADP, right? Active transport is broken down into two kinds. We call it a primary transport because that's when ATP is immediately used to move something against its concentration gradient. So, for example, let me just kind of get this going. Presley, stop. So if you have a cell, and on the inside of the cell, you have a high concentration of hydrogen ions. On the outside of the cell, you have a low concentration of ions. If this was passive transport, things go down the gradient, so they would go from the inside to the outside. But in an active transport, you move things against the concentration gradient. So in an active, I'd forcing hydrogen in here, okay, or reverse, one or the other. Let me actually, it doesn't really matter. Hydrogen would be more concentrated depending on what kind of cell on the outside. But anyways, in an active, I'm going to force these hydrogens back in. They don't want to be all clustered here. So I'm going to force the hydrogens in against their will, basically. That's going to have to use your proton pump. Okay. I'm going to have to force a proton pump in, which is then going to have to use ATP energy. Then what you end up getting is hydrogen ions on the opposite side of the membrane. Like I said, I really need to flip these where I have outside and inside. Let me just do that real quick. Then you're going to be forcing the hydrogen ions where they didn't want to go, and they naturally want to get away. No molecules want to be crammed up. That's just the basis of diffusion, right? Going down a gradient. So all of a sudden you have all these hydrogen ions forced where they don't want to be, okay? So you forced hydrogens in. These hydrogens don't want to be there, so now you're going to force the hydrogen, or the hydrogens are naturally going to want to go back out. So when the hydrogens go back out, or in this case, I mean in, when the hydrogens actually move back, they passively diffuse at this point. So here in the proton pump, first of all, if it's called a pump, that lets you know that energy had to be used. ATP was used. A pump lets you know it is some kind of active transport. So in this pump, this would be called primary active transport because we had to use ATP. But when the hydrogen passively diffuses back through, it also ends up taking with it sucrose or sugar. So then the sugar gets to move back as well. Sugar doesn't get to just move. It has to be facilitated in some form or another. It's too large of a molecule. So as the hydrogen diffuses, he kind of pulls sucrose back with it. That's what we call a secondary active transport. It is moving molecules with the ultimate, back at primary, use of ATP, but not the immediate use of ATP. All right, so so we call it a co-transport because hydrogen moves, but because it moves, it transports sucrose as well. Okay, Lauren, let me know if that helps. What type of functions do peripheral proteins have? It seems like most of them only apply to integral ones. Good job, Patrick, for answering that question. Yes, guys, your worksheet is due tomorrow because it's the test day. Worksheets are always due on test day. All right, so peripheral proteins. So yeah, you either have your integral, which the type is transmembrane, or you have your peripheral. All right, those are your only real types. All right, good job, Lauren. Those are your only types of proteins. The transmembrane literally go across both ways. So a transmembrane protein is going to, one, either be some kind of channel, some kind of pump, or some kind of receptor, right? Your integral proteins. So an integrated protein can be integrated on the inside partly, or it can be integrated on the outside. What I mean by this is either you have a protein that is integrated but exposed on the top of the cell, or integrated but exposed to the inside of the cell. All right. Either way, an integral protein can, if it is difficult for an integral protein to act as an intracellular, like a receptor on its own. If you have an integral protein acting as a receptor or some kind of gateway, he's actually going to have to be connected to a transmembrane in some way. So, Megan, yes, you're thinking, oh, you said integral. Yeah, in the peripheral. I'm getting there. So they all kind of go back to kind of having to interact with the transmembrane somehow. Now, what you can see is an integral protein attached on the outside of the cell being attached to a glycoprotein as a cell-to-cell recognition. Remember, glycoproteins and glycolipids are our cells' way of talking to each other and shaking hands. All right. Then your peripheral proteins. So peripheral proteins are either on the outside or on the inside, but they never actually cross into the membrane at all. Peripheral proteins can, a lot of times, we would qualify your protein kinases as a peripheral protein because a lot of them actually do act that way. Peripheral proteins are very mobile. So because they're not integrated into the membrane in any way, they can actually migrate around the outside of your cell and the inside of your cell really well. That's why your G protein is a peripheral protein. It gets to actually shift from that one transmembrane receptor to the integrated membrane adenyl cyclase. All right, so peripherals do have to talk to integrals. The peripherals have to talk to transmembranes. But because they're mobile, it makes them very necessary. Okay, number 11 on the worksheet. What molecules will use a specialized... Tatum, aquaporin, aqua as in water. Back in the 90s, we finally figured out that water we always knew water could not pass through the cell membrane just by itself. We realized that. There was no way for water to get past those fatty acid chain tails. So we couldn't they couldn't explain it. So it wasn't until in the 90s when they actually discovered the protein channel that allows water to pass. They are small little specialized transmembrane proteins that they called aquaporins. They are scattered throughout your entire cell membrane, and every single one of them that allow water to go in and out, in and out, in and out freely. Okay, that's all an aquaporin is. All right, number 11 on the worksheet. I've got to pull it up to be able to look at it, so hold on. Number 11, Kriege, it says, in the intestine, sodium ions and an amino acid bind to the same transport protein that moves the two substances in the same direction. This is an example of what type of transport. Anytime you're having to move two things, you're talking about a co-transport. So it's the same exact example as the hydrogen and the sucrose from the notes. So the other thing that you can kind of always notice how in both those examples an ion, hydrogen, and sodium are being used. An ion usually is first pumped against its gradient, meaning an active transport. And then the ion diffuses back down its gradient, meaning a secondary transport. In this case, co-transport. So you can answer that kind of question both ways. If you said it was a secondary transport or a co-transport, I accept either, because when you're dealing with ions, it's going to be a secondary at that point because the ion got pushed out against its will. Okay, let me check this. All right, Olivia, you said signal transduction pathway and cascade. I'm feeling that's kind of a big one. So we'll definitely go into that one here in just a second. Dynein, nexine, and kinesin. Those are just motor proteins they're the specific names. I don't really need you to know them anymore. They're just, I haven't modified some of my worksheets in a few years. So dynein, nexing, kinesin, I really am not too stressed about that, Megan. So you can kind of skip that. Hold on, let me look real quick. Megan, do I have that as a specific question on the worksheet? Let me look. If I have it, I feel like I don't because I redid this worksheet just the other day. Is it on the test review, Megan? Oh, okay, it's on the test review. Yeah, those are specific terms that you'll see under some of the just vocab because it's part of the chapter and it's part of the Prezi. But if I didn't stress it, don't worry about it. Really. If I don't stress it in class, it's not anything that you need to worry about. What molecules need a carrier protein to be moved across the membrane? Anything that cannot freely pass through the membrane has to have a carrier protein, Tatum. So you're talking ions and large molecules. Okay. All right. Does endocytosis and exocytosis use vesicles or lysosomes? Lysosomes are a type of vesicle. Okay. Both of those are created from the Golgi. A lysosome is just a specialized vesicle that has hydrolytic enzymes on the inside, whose job is to bind with another vesicle to clean up whatever was inside of it. Because a vesicle could be created inside the cell and has broken down cell parts that need to be destroyed. So it fuses with the lysosome, creating what we call a phagosome. Or you can take something into the cell through endocytosis, create a vesicle. That could have bacteria in it and gets broken down. So both of them are vesicles, but you only talk about the lysosome part if it has hydrolytic enzymes in it. And its job is to fuse with the other vesicles. Colin, I'm about to do transduction. That's like a big, big one. So I'm just trying to get some of these smaller questions out of the way, and I'll go into that one, okay? Kriege, what do you want? Big facts. Like, I don't know. That's a general kind of question. Yeah, I would study your worksheet. Yeah, for that one. Okay. All right. Well, no other kind of smaller questions, then I'll get into the big, big one, which is the signal transduction. All right. So I'm just kind of drawing on a notepad. All right. So to do the signal transduction, I would definitely say be able to write this out just a little bit and like draw it out yourself. It doesn't have to be pretty, or be able to look at the picture and walk yourself through it. Wait, back to Tatum's first question. What molecules will actually use that aquaporin? Water. Aqua. Water. It's a porin. It's a pour for water only. That's it. Can you just show us the test? Thank you, Patrick Water Blake. No. Okay. And I know that last little video or whatever, I accidentally had the test up. That was an old test, y'all. I'm sure half of y'all probably froze the screen trying to study those questions for a split second. Ha. Okay, so let me kind of draw the signal transduction first. All right, so when it comes to signal transduction, remember you've got quite a few different proteins. You actually have all three represented in this one picture. Wink, 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 wink. All three proteins are represented in a signal transduction pathway. Wink, wink. Okay, so you first have the transmembrane, which is going to be your receptor protein. And keep in mind, guys, signal transduction happens in all of your cells. I just show you one example of the liver cell, just because it's the most identified. It was one of the first identified, actually, so that's why they use it. But signal transductions happen in all cells. All right, so you've got a receptor protein. It's got a spot for the receptor. Keep a general term in mind, allosteric. An allosteric just means a different site. That's literally what that term means. All right, so in a transmembrane protein, its job is to receive some kind of chemical that we would call ligand, right? Any kind of molecule that's going to bind to a receptor. But the special component about this guy is on the inside, that transmembrane has a peripheral protein attached to it called a G protein. See, you can tell it's peripheral. It is not integrated, but it is touching this other membrane. This other protein, because it's going to actually have a chemical reaction between it. As soon as that ligand binds to that receptor, it's going to activate that G protein. When that binds, so I'm going to show binding, then all of a sudden that G protein becomes activated, and it gets activated with a GTP. GTP is just the cousin to ATP. It's still energy. ATP stands for adenosine triphosphate. GTP is guanosine triphosphate. Ooh. Okay. So you've got energy. So ligand bound. G protein activate. Okay. Kind of sound like I'm a transformer in a minute. Right. So GTP, it's now activated. This protein's all excited. Remember, peripheral proteins are mobile proteins. So what's it going to do? Hop, skip, and jump that way. Because what's over here is an integrated protein called adenyl cyclase. Okay, integrated protein, adenyl cyclase. Activated G protein is going to go whoosh, and move right over here. So all of a sudden, he's shifted. The G protein, then, when it comes in contact with adenyl cyclase, loses a phosphate. Why? Because he's going to activate adenyl cyclase. And to do that, it's got to use up his energy. So over here, that GTP has now been converted into GDP. It's lost its energy. And so once he's activated adenyl cyclase, what you would see is that GDP has done his job. So what will he do? He'll go back over here and just sit and wait. Sit and wait for another ligand to activate him so he can shroop, deactivate, go back, activate, shroop, deactivate, go back. So now that a phosphate's been released over here, adenyl cyclase, that's where this protein passes on a phosphate, creates energy again, but now in the form of ATP. What? How did ATP get created? Because what just got thrown off at GDP? A phosphate. So because of adenyl cyclase, ATP gets created. ATP is immediately used because ATP's job is to create this guy, one of the most important guys in a signal transduction pathway, CAMP. All CAMP means is it is another energy molecule. It is cyclic adenosine monophosphate. So instead of having 2 phosphates, instead of having 3 phosphates, it only has 1. And it's cyclic because it actually bonds with itself twice. So it kind of completes the bonding. CAMP is what we call a secondary messenger. What does that mean? It is the internal messenger for the chemical signal that happened up here. This is the original signal that started all of this. But as soon as you get inside the cell, any kind of cell membrane reception, it's got to somehow transduce the signal here. So that's what CAMP is doing. It's passing on the rest of the message inside the cell. So this is, you can kind of think of it, all of this happened because of the ligand. But if you want anything else to happen inside your cell, you got to have someone else do the rest of the job for you because you have officially lost all of your energy. So what do? You charge this guy to finish the job. So CAMP, secondary messenger, found in all your signal transduction pathways. Now here's the thing. He's not the only one. CAMP is not the only molecule that can do this. It's just the most popular one found in our body. So CAMP's job is to then cause a signal cascade. All a signal cascade is you activate or deactivate molecules. How do you know which one you're doing? It's just because that's what we found and have discovered. I don't need you to know them. I don't know them. All you're needing to understand is that a cascade happens and what that looks like. So over here, you might deactivate one thing. Over here, you might activate one thing. And then that activation causes another and another and another, right? If we are specifically talking about one particular example, which one I needed you to know, the liver. The liver stores what? Glycogen. What is glycogen? Starch. What's that going to do for us? Break into sugar. What's sugar going to do for us? Give us energy. Right? So, CAMP ultimately stops the synthesis of glycogen in your liver and says, hey, liver, stop holding on to this glycogen. We need it right now because I need this person to run a marathon now, and they didn't plan on it. Now they're stressed, right? I need you to start releasing all this glycogen as sugar. Sugar is then going to leave the liver cell. As sugar leaves the liver cell, it's released into your bloodstream, travels, goes to your cells. Sugar becomes part of cellular respiration. We haven't touched that yet. And it becomes energy. Okay. All right. If you can kind of, first of all, draw the three kinds of membrane, three kind of proteins, transmembrane, integral, and peripheral. In the signal transduction, show that one gets activated, activates the other one. That ultimately causes activation of your secondary messenger, CAMP, which then causes a cascade inside your cell. The purpose of signal transduction pathways is for your cells to receive a signal, cause some kind of chemical reaction inside the cell to get the cell to give you some kind of response. What that response is depends on whatever cell it is. It can be as simple as a liver, I need you to release the sugar. It could be the hypothalamic cell saying, I need you to make this hormone. It could be your testes or ovaries saying, I need you to start making some sperm or eggs. It can be anything, okay? Cell junctions, yes. So much GDP nowadays with bio and macro sheesh. Oh, GDP. Slightly different. In the Bozeman video, he uses ATP instead of GTP. Can we use either one? He uses ATP attached to your G protein. Bitter. I mean, technically that's wrong. But yeah, the G protein is just that. It's activated with GTP. So I can't see why he would use ATP. All right. That's just weird. No, it's actually activated with GTP. I'll definitely look at his video and like send him an email or something why he would use that. Okay. Cell junctions. Okay. Guys, all a cell junction is connections between two epithelial cells. All right. It's how cells get molecules and chemicals to each other quickly. And I say epithelial cells. It's muscle cells. It's any of your cells that are connected to each other. So these are not junctions, are not something you're going to see between red blood cells and white blood cells. They aren't attached. This is only cells that have some kind of layering effect to them. Muscle cells, skin cells, right? So here's what you got. You've got two skin cells, two epithelial cells. Maybe these are two muscle cells. Either way, they need to be able to talk to each other and get chemicals to each other. So you do that through either open or closed junctions. Those are your two categories. Okay, open or closed. All the junctions we talked about were animal cells, except for one. One opened one was for a plant. It was called a plasmo for cytoplasm desmata opening. So plasma desmata is how plant cells are connected. That's why the big argument says that plant cells actually aren't multicellular. They're one big fat cell. So that's kind of interesting. Oh, and someone's texting me. Okay. All right. So the plasma desmata, it's an open one, but that's for plants. Here's what I need to know for animals. Animals have an open one called gap. It literally is a connection channel between both sides. Why would you need that? So that molecules can flow inside and outside from one cell to the other. Gap junctions necessary in all connected cells. This is the only way you can transduce any kind of molecule or chemical or electrical signal quickly through some kind of gap. So this is going to be skin cells. This is going to be muscle cells. Okay. The other two are called closed. There's desmosomes and tight junctions. Again, all junctions are ways in which layered cells communicate with each other, get chemicals to each other. Desmosome are actually anchors. One side of the cell is literally anchored to the other side. It's anchored to the other side, to the other side, and the other side like this. Think of them like sutures. These microtubules literally are embedded from one side to the other, just across your cells. Okay? This problems in people's desmosomes inside their epithelial tissue is unfortunately also we all will have problems with our desmosomes as we get older. Desmosomes start to malfunction as we start to age. That's why older people start to have what's called like fragile skin, and they bruise really easy. They cut really easy. They get what we know as thin skin. Desmosomes is part of that problem. They don't have good anchoring anymore. And then you get what's called a tight junction. A tight junction doesn't actually go into the cells. It's formed on the outside of cells' extracellular matrix right here. A tight junction is like a barricade. So desmosomes allow things to flow between. In between. Desmosomes are anchored, but watch what happens if molecules flow between cells, just in between their membranes. Okay? If these little molecules are in between your cells, they can freely get between the desmosomes, guys. Those aren't completely barricaded, but they cannot get past a tight junction. And notice where the tight junctions are. A tight junction's at the end, so that any molecules can't actually leak out of that tissue. You don't want certain molecules leaking out of your intestinal tissue into your surrounding peritoneal tissue. Like, that would be bad. Okay, so a tight junction, think of it as literally, it's a fence. Nothing gets to get by in between the cells. Okay, this isn't, nothing can get by from the inside and outside of my cells. It's nothing can get by in between your cells. Okay, it's a fence. Let's see. Do we need to know autocrine, paracrine, and hormones? Yes, that's how cells talk to each other. Autocrine are chemicals that affect the same cell that released them. Paracrine are hormones or chemicals that get released and affect a nearby cell. Hormones travel through the blood. So cancer cells are very autocrine. Paracrine cells, that's literally pretty much, that's every cell in your body. They all talk to each other. They all release not hormones, but chemicals and ligands and talk to each other quickly. Hormones, that's exactly what it sounds like. It's either being released from your brain or other organs in your body. A hormone's job is to affect some other cell or organ or tissue farther away in the body. So it's got to be able to travel through the bloodstream to get there. Okay. How do you tell if it is secondary active transport if it doesn't mention ATP? Not sure what you mean, Isabel. Like in the question, the fact that it says the sodium and the sucrose, like how do you know that's the sodium and sucrose or whatever was secondary active transport? It's just an example, just like the hydrogen and sucrose one. You would just it would just be something you'd have memorized. The secondary active transport still goes against the gradient, while passive does not. Thank you, T, for going ahead and answering that. Sorry, I'm still eating some fries.
Give me some fries too, please.
If y'all didn't know, I have my daughter in my bed with me. What evolutionary advantage does a eukaryotic cell have over a prokaryotic cell? Think about what a eukaryotic cell has. Organization, compartmentalization. The fact that inside of our cells, we have so many different organelles that have such a specific job that we can also separate out all of our processes and make our cells run more efficient in a way. And I know really, and you kind of think, but wait, a bacteria is so simple. And yes, they really are. And that makes them efficient in that regard. But when we compartmentalize, it allows us to control the energy use. And we get to prioritize certain molecules and certain processes over others when we have all these separate membraned organelles that a bacteria does not. Question 11. Okay, I'll look at it. 15. All right. Excuse me while I'm still eating my fries. I said I didn't have dinner. Number 11, number 15. Let me see. I'm pulling it up. All right. Yeah, number 11. Isabel, I went over that earlier. Somebody asked that earlier. You know that one, because it's when they're having to go against, when molecules are having to go against their gradient. And in this case, a sodium ion has been pushed against its gradient first. So that was its primary active transport. And then when it's pulling molecules back along with it down the gradient, we know that that's called secondary active transport. All right, number 15. The mammalian hormone insulin binds to a receptor on the outside surface of the cell and initiates a response. Your insulin receptors, it's just directly out of our notes. Insulin binds to your protein kinase receptors. We talked about ion channels, right? Your ion gated channels would have some kind of ligand that opens up the gate for sodiums and potassiums and calciums and stuff to flow through. Well, a protein kinase accepts insulin. Now, I guess I can kind of draw this. The protein kinase and the exact how it works, I'm not too concerned with. I want you to know the signal transduction one, for the liver cells and adrenaline, epinephrine. But for a protein kinase, you have a different kind of membrane protein. So protein kinases, this is just the particular one for insulin, okay? What happens is your insulin ligand binds, okay? And when it binds, just like the other one in your liver cell, when adrenaline binds, it activates that protein. But in this case, the activation doesn't have a linked G protein. It actually has phosphates. So when it gets activated, inorganic phosphates bind. When those phosphates bind, that protein is now activated, which then those phosphates are going to now cause an effect on another protein kinase inside. Okay, so protein kinases were just one of the other kinds of membrane receptors mentioned. And insulin is just the particular ligand that binds to a protein kinase. It's just an example.
Can I draw something?
No, ma'am, you cannot. I got ice cream on my paper. I'm sorry. Your worksheet's due tomorrow, that paper. I'm sorry. Hormones bind to protein receptors, yes? Protein kinases or membrane sense it's insulin. I don't know what you mean, Lauren. Protein kinase or membrane. I'm not sure what you mean. A protein kinase can be a membrane receptor, and it can be an enzyme protein acting inside your cell. Protein kinases are just a type of protein. You can find them anywhere. So its particular form of a protein kinase inside the membrane binds insulin. But then protein kinases inside the cytoplasm just act as an intracellular protein. Not gonna lie, still lost on 11. Okay. All right. I don't know what other way I can explain it. I guess I can draw 11 one more time. Patrick, I think it's because it uses sodium and protein, so I think it's talking about a sodium-potassium pump. Okay, time out. Okay, Patrick, Colin, and Blake, I don't know what's going on. I answered the 11 question talking about secondary active transport. Just now I was answering number 15 for Lauren, talking about protein kinases and insulin. So this little drawing was for number 15. An insulin binding to a protein kinase protein. Like that membrane receptor is called a protein kinase. I think Lauren is asking if the answer is a membrane. Oh. No, again, Blake, your thing, because I think Lauren is asking if the answer is a membrane or a protein kinase on 15. Insulin can't just bind to a membrane. There's no spot for it to bind to membranes. That's why we use proteins. And insulin specifically binds to protein kinases. They are one of the 3 types of membrane receptors I wanted you to know. Ion-gated channels, only for ions. Protein kinases, the example I gave you was insulin. G-protein linked adrenaline. Okay, Patrick, I'll try and do this one more time. One more. Okay. Alrighty. Do. I'm doing the transport one real quick. Okay. So for transport, secondary, primary, it doesn't matter. Active transport. An active transport is going to happen because you're pushing something against where it wants to go. So it's called going against its gradient, guys. All a gradient means is a change in concentration from one side to the other. You can talk about the gradient of your driveway. Do you have a high gradient or a low gradient? High, low, high, low. So if you're going against your gradient, you don't want to do that. It makes life hard. And inside your cells, you don't want to have to do that because you're going to have to require energy to do that. Okay, Laura, I was reading yours. Okay, so if you're going against the gradient, that means high to low concentration. Diffusion, which is passive, is, or sorry. Diffusion is a high to a low. But in this case, I'm going to force these guys out. Yes. I want to force these hydrogen ions against their gradient where they don't want to go. They don't want to be crowded. So to do that, I'm going to use my protein. In this case, specifically, it's called a proton pump. It's a protein. It's a protein pump, but we can call it a proton pump because that's all hydrogen is. It's just a proton. So, but anytime you see the word pump lets you know that it's going to be active transport. So I'm going to force it. It doesn't like it. This used ATP, primary transport. Then they want to flow back in. But when they do, they're going to drag sugar with them. So sugar's like, yay, I get to go in the cell. Okay. This is co-transport. Anytime an ion, any ion, drags with it a larger molecule, we call that co-transport, aka secondary active transport. All right. Okay, Lauren, insulin can't just bind to a membrane. The kinase is in the membrane and grabs that puppy. Thank you, Patrick. Okay. Licia, can we go over 23 and 26? Okay. Blake, Presley, stop. 23 and 26. How does the plasma membrane of a cell compare with the membrane in the U-shaped tube? All that is, that U-shaped tube, it's just an example. It's a piece of equipment that we can use that actually tries to give us a good representation inside the lab of what a cell membrane would act like. Because in the middle of the bottom of that tube, they actually have created a semi-permeable membrane. So they use this kind of polymer. It's like, I don't even know that's gelatinous, but they use a polymer that's in the middle. So you can kind of think of it as a, just like a piece of plastic with like foam in the middle of it. And that foam, in a way, only lets certain things go one side or the other. So that U-tube, one side can represent the inside of the cell, and the other side represents the outside of the cell. And so it just allows for a scientist to experiment with a cell membrane in a lab without actually having to utilize a cell, because it's representing that semi-permeable membrane. And the top of the tube is actually open. So you can actually put plungers in it and change the pressure. So you can treat it as if pressure was being added to it. It was a turgid plant cell. You can remove pressure as if it was a flaccid plant cell. You know, kind of change the pressure on it. All right, 26. What happens to the pressure inside a cell that is placed in a hypertonic solution? All right. All right, so you draw kind of like your beaker, yay. Draw your cell. If a cell is placed in a hypertonic solution, so I'm going to say that that's 75% out here of solute concentration, that's, 20% solute concentration. So all of a sudden, water is going to want to leave because, right, high to low, there's not a lot of water here, so it wants to run where there isn't a lot of water. And unfortunately, whenever a plant loses any kind of water, that pressure inside the cell is now going to decrease. Think about a flaccid cell, a flaccid plant. It's wilted. It doesn't have any pressure pushing on its cell walls anymore. Think about an animal cell. All of a sudden, that animal cell is very soft, very malleable. It's crenated. There isn't much pressure from the inside acting on it anymore. It's dead. It's dying. All right. What else? Other questions from the worksheet. Did I skip anybody's question? I don't think I did. Lauren, are you good? Patrick, are you good? Cooper, are you good? Presley, where's my... Oh, there it is. All right, let me see. Let me know if there's something else I didn't. Number 25. Is number 25? Yes, okay. Does osmosis occur if a cell is placed in an isotonic solution? Yes. Guys, water can never stop moving. It doesn't matter if it's isotonic. The difference is there's no net flow. One side of the cell is not gaining more than the other, guys. Net means you have extra. You have taken everything else away and you still have something left. There's no net flow. But water never stops moving. It can't stop moving. If water ever stopped moving, you are dead. This is Sarah B. Please move the test to Tuesday. No. Test is tomorrow. Yeah. What other questions? I don't think I missed anybody else's questions up there. Strongly disagree, Sarah. The reality is there's always going to be one person that wants to move. The other person's like, no, I already studied. I already spent time. I'm not going to have time to study again. Or, no, that's the day of my calculus test or my micro test or da-da-da-da. So, no. All right. When glycolipids shake hands, is the cell recognitions? Yes. Glycolipids and glycoproteins are cells talking to each other. That's why you have to take anti-rejection meds when you're on a transplant because your glycolipids and glycoproteins are literally your fingerprints of your cell. So for any reason, if your fingerprints don't match and they can't shake hands, they're going to send an immune response and kill you. Number 5, is it because of high concentration of cholesterol? And then number 27, number 5. Which of the following is one of the ways that the membranes of winter wheat, that's a plant, are able to remain fluid when it's extremely cold? Okay. Cholesterol, yes, but your first go-to answer for that is actually going to be unsaturated fatty acids, because cholesterol will help maintain fluidity as well, but only to a certain extent. Too much cholesterol can actually make cell membranes rigid and stiff. So your cells will first add unsaturated fats. So if you have saturated fats, right, they're all straight. But the second you start incorporating unsaturated fats, the kinking causes more space. More space means more fluidity. Okay. So you want this first. Any of your cells, whether they're plant or animal cells, are going to create unsaturated fatty acids first, and then add cholesterol as needed. Okay, number 27. Patrick, let me know if that answered your question. What causes a plant to wilt? Well, think about it. If a plant wilts, that's because it doesn't have any pressure. And if it doesn't have any pressure, we know that that's directly linked to the amount of water it has inside of it. So if a plant's central vacuole isn't swollen, pushing against the cell wall, making it nice, stiff, and turgid, that means the central vacuole is not full of water, right? It's smaller. And so therefore the plant itself has a lower concentration of water in its vacuole. So we would need to place it in some kind of hypotonic solution, right? So that'd be like distilled water, most hypotonic solution, so that it can take water back up into its central vacuole and swell and put that pressure back on the cell walls to stand back up. Let me know if that answered your question. A plant will wilt in isotonic as well, right? Because it's only turgid in hypotonic. Yeah, isotonic, it becomes what we call flaccid. So yeah, it starts to kind of wilt a little bit. It's only turgid in the hypotonic. All right, 27. Oh, sorry, that was 27. Lauren, can you explain 10? Would it be transmembrane or channel? A protein that forms an ion channel through a membrane is most likely, what was your question? A channel protein is a type of transmembrane protein. They're like one and the same. Because it's an ion channel, it's a kind of channel protein, which we would just qualify as transmembrane because it's acting on both sides of the cell. Okay. What are you doing?
Just stretching.
Presley. Walker 21. Ions can travel through the cell membrane by use of. Hey, Lauren, you can answer that question for Walker. He wants answer number 21. Walker, we kind of just answered it. It was kind of the question Lauren just asked. So, okay. Hey, Patrick, it's not just active. They're first going to be able to go through an ion channel. Transmembrane, correct, Patrick? It's if they're being forced against their concentration gradient, we would use a protein pump. But ions can just move through the membrane passively through an ion channel. So just kind of know both. Good job, Lauren. All right. What else? Oh my God, I can feel my eyelids just getting heavy. Oh, hush. What are you having for dinner? I didn't have dinner. I'm literally eating French fries from Whataburger. That's it. That's my dinner. But you know what? I don't freaking care because Whataburger fries are like the best.
And she's sharing them with me.
I'm not sharing them with you anymore.
Yes.
Can you please explain bulk transport? It moves things in bulk. I'm sorry. I'm getting tired. So my snarkiness is coming out. Okay, hold on. Hold on, Tim. All bulk transport is a cell is needing to take in a high quantity or get rid of a high quantity of some kind of molecule. So it's going to form a vesicle around it, either inside or outside. It's going to take it in. If it's a solid object, phagocytosis. If it's some kind of dissolved molecule in a liquid, then it's pinocytosis, drinking. Or it's going to exocytosis, push things out. So you would see that as pino, any kind of dissolved hormone. It would take it in one big gulp if it needed to. You would see it phago if it's eating, like a macrophage eats a bacteria. Okay, that's phago. So it's taking in a solid object. And then exocytosis would be after it ate the bacteria, it fused that vesicle with the lysosome, creating a secondary lysosome, like a phagosome. And the lysosomes all chop, creating little tiny bacteria nothings. And then it's going to push that vesicle back to the cell membrane and shoot it out as waste. And you get rid of it through multiple ways. What molecules need a carrier protein to cross? Anything that's not small, essentially. Yes. Gas, O2 and CO2, those are the only things that get to pass through your membrane without any kind of concern. Water has to have a protein channel that just kind of flows straight through. Small lipids. Small lipids can, and when I see small lipids, can pass through your membrane. So we're talking small beginnings of a cholesterol molecule or something. They can pass straight through your membrane. But other than that, gases are the only thing that can pass. Everything else has to have a protein in some form or fashion, whether it's a protein channel, just going straight through, like facilitated diffusion stuff. It's a gated channel for ions, or it's a pump or some kind of receptor. Everybody's got to have help. Carrier proteins help larger molecules cross with passive transport like glucose, correct? Help larger molecules cross with passive transport like glucose. Isabel, yes, but you need to be, that would be more specific. You'd have to say it's facilitated diffusion, right? Because it is a type of passive transport. As long as no energy is being needed, we call it facilitated diffusion because a protein is being used. Anytime a protein gets used and it's still passive, we call it facilitated diffusion. Number 14, endocytosis. Thank you. Number 14, phagocytosis. Ooh, okay. Oh, y'all are both asking the same question. Number 14, endophago. First of all, phago is a type of endo, just so y'all know that. Phago is a type of endo. Plasma membrane engulfs large particles. Y'all can say either one. You can call it endo, or you can call it phago. I'm okay. Either way works. Anything else? I feel like we crammed a lot of stuff in 1 hour. Patrick. Yeah, I agree with T. Not a chance. Y'all might not know, I have a couple other side jobs that I do besides teaching. And so, yes, please kick him. So I was doing one of those literally all day yesterday. So I didn't finish till 10 o'clock last night with that one. So, and then we have Sundays are church days and family days. I don't really do anything on Sundays. So be happy I'm doing this. Okay. I'll start grading them tomorrow. Worried about the fill in blank. Will it be a lot about the organelles? I mean, it's not all organelles by any means. It's organelles, and maybe it's still just a membrane protein question. The signal transduction part, I couldn't really see part. It's probably not going to be part of the fill in the blank. She's wasting our time eating fries, not even grading my teacher. Alicia, Alicia, there is always fill in the blank. Your tests are always a combination of everything. Well, I say everything. You will always have multiple choice. You will always have fill in the blank, and you will always have short answer. Every once in a while, I'll throw in matching. I never do true false. But you will always have fill in the blank, multiple choice, short answer. Is there a bonus? There is always some, well, I say not a bonus. Your bonuses are incorporated. They are the AP questions. However many I choose, I usually only put 1 or 2. That's like it. But those this time are only going to go back to counting as the 1 point, right? And the bonus question is, I haven't added one on there right now. I'll see if I want to add one. I'm not real sure. Am I screwed? No, you're never screwed. Never. You're good. Do we need to know the structures of the organelles or mainly just the functions? General structure would be nice. What I mean by structure is like the parts of the endomembrane system. Alicia, I'll answer yours too. A 0 is a good grade. It is. It's better than a -50. Exactly. Okay. Yes. So first, know who's part of your endomembrane system. What that means is what formed from the inner layer of the cell membrane. So the cell membrane invaginated to make the nucleus. The nucleus is attached to your rough ER. The rough ER is attached to your smooth ER. The Golgi then also formed from the inside. Golgis create vesicles and lysosomes, which are simply just a type of vesicle. Okay. So like knowing your endomembrane system, if you know that they're the endomembrane system, that automatically tells you that those are flat membranes. Kind of makes sense. That is their structure. They are flat membranes. Now, unique thing about the Golgi, because the Golgi is our post office and kind of sits close to the cell membrane, it has what we call 2 faces. It has a cis face and a trans face. The cis accepts proteins and lipids from the ERs. The trans transports outside the cell. Other than that, I don't need you to know the chloroplast structure or the mitochondria structure or anything like that. Just being able to maybe describe that the endomembranes are flat membraned or thin membrane sacs and stuff. That be kind of the only structure. The functions are the most important, Tatum. Extracellular matrix, yes. Chart quizzes in pen. This calculus test is going to be killer. I couldn't tell you a lick about what we're doing in econ, and there's not a chance I'm wasting bio time figuring that out. Okay. Not sure what that chart quizzes in pen thing is, but Alicia, let me answer your extracellular matrix question. So all of our cells that are connective, again, Connective meaning like epithelial and muscle. This isn't going to be red blood cells or white blood cells. This is only things that have to be joined together. So on the outside of your phospholipid bilayer, you're going to have literally extra kinds of proteins kind of scattered around. Think of them as like little pieces of rebar holding things together. Okay. That protein is called a proteoglycan. Okay. This like rebar that's kind of stretched across. It's called a proteoglycan. So protein sugar. That's all it is. So it's kind of, think of that as the fuzzy pieces of your Velcro. So the fuzzy sides are this stuff. Then your cells have another protein that sticks up and kind of hook onto that proteoglycan. That's that rough part of your Velcro. Because if you look at them, they really are like tiny little swivel pieces of hooks of plastic. Those are called integrins. So again, it's just these are all just proteins. That's all your extracellular matrix is. I haven't stressed it enough. Proteins are literally everything to you. Every component to you and your cells has some kind of protein. Holding it together, making it work. Yeah. So your extracellular matrix is how your connective tissues, all right, how they stay together. So your junctions is how they communicate and keep molecules where they're supposed to be. The extracellular matrix is how they join together and stay together. Because if they didn't hook on together, basically you'd have this layer and this layer of cells laying on top of each other, constantly like shifting and moving and tearing. When your extracellular matrix is deformed, you have that condition I mentioned in class, the EB, epidermodysplasia bullosa, where your skin literally tears. Not bruised, not cut. It tears from birth. So the act of bumping or anything like that, those individuals will get blisters, and those blisters will just peel their skin off. So if we could explain this stuff but not have proper terminology for each thing, will be okay. Example, those big words you use for the ECM. Some of those vocab, like proteoglycan and integrin, I would see those not necessarily being the fill in the blank words. Maybe I flip it, you know, like there are some words that are more difficult, but we have stressed, right? You know, like signal cascade, camp, like some of those words we stressed, but like proteoglycan and integrin, I haven't stressed, but I have stressed ECM. So maybe I flip it, you know, maybe on the fill in the blank, I talk about proteoglycan and integrins are proteins found as part of what necessary structure in the cell or as part of the cell. And you would fill in ECM. See that? Like I would kind of flip that vocab, you know, what I've stressed would be what you would fill in. What I haven't necessarily stressed, yes, would they still be part of the question? Of course, because I still want you to know them. But you wouldn't have to necessarily be worried about trying to remember how to spell them, if that helps. Okay. Alrighty. Presley's over here creeping. Mm-hmm. Mm-hmm. All right. Anything else? Hmm. No. It is past my bedtime too, T. I think we all need to go to bed. I think y'all studied what y'all can study. A part of the desmosome junctions. No. The night is still young. No. Desmosome junctions, ECMs, two totally different components. What you're seeing is, can they both have similar kind of effects if they're malfunctioned? Yes, they can. Desmosomes anchor cells to each other beside them. ECM anchor them on top. So if either one of those fail, you're going to have tearing, epithelial malfunctions, but they are not the same. Anything else? No rip, Megan, no rip. Lase in our signal transduction pathway. No. All phospholase is the enzyme that adds the phosphates. If anything, next unit, I'm just going to make sure y'all can stress the term phosphorylation, which is the actual process of adding phosphates. I don't need you to worry about the enzyme because hopefully the enzyme name just kind of tells you already. Because anytime you add ASE to the end of the word, that is an enzyme. So the fact that it says phospholase lets you know it's having something to do with phosphates. I want you to know phosphorylation, but I haven't talked about that yet. Why? Because we're going to talk about it next unit. Major rip. I'd kill to be playing on an iPad, not strung out on Red Bull, stressing her. Yes. Presley, what are you playing?
I'm not playing it.
Show them. What do you have? This is the crap my child watches. YouTube instructional videos on how to play a toy. Rather than playing a toy, how to play with a toy. This is parent winning. Like, I'm winning at this parenthood. Awesome. Like, I'm the best. I'm back. So sad. Krish, did you just miss the entire thing? Because we're actually wrapping up. Yeah, like we're actually finished. Guys, I'm going to post the video. Oh, I know. T, trust me. They accidentally found some last year we saw in their YouTube history. That was weird. Oh my gosh. We'll just say SimCity. Some people aren't very nice on those things.
Mommy,
Okay. Yeah, I'm going to post this on Canvas so y'all can watch it if you need to. So you're not totally screwed, Krish. Okay. All right. Are we good? Yeah. I'm going to finish eating my fries and then go to bed.
With me.
Nope.
Yep. You're going to bed with me.
John just got clapped. No chance, John. It's a rip. All right. If nothing else, T, you were right. Oh, Colin, have you run into some of those channels before? All right, guys. If there's nothing else, then I am. All right. Bye, guys. Yep. See you later. Wait. Okay. Walker, what? Walker, what? You better type fast. Mm-hmm. You're welcome, guys. Walker all of a sudden is saying wait, so he better type his question. What's the process used by white blood cells to digest stuff? Phagocytosis. They engulf things through endocytosis, specifically a type of endocytosis called phago. Phago is when they take in something solid. Say hi, Blakey Boo Boo.
Hi.
All right. Walker, just let me know that you're good. Ugh, cowboys. Ew. All right, bye, guys.